Hello and welcome, dear students. Today we are going to discuss the topic non-Mendelian inheritance, codominance, multiple allelism and incomplete dominance. The main objectives of this module are to know about the basic of Mendelian genetics, to understand the Mendelian inheritance versus non-Mendelian inheritance, to know about the concept of multiple alleles, to know about the phenomena of codominance and incomplete dominance, and finally to understand the inheritance of blood groups in humans. Dear students, let's first begin with the introduction. Genetic principles are the rules or standards governing biological phenomena of heredity, the transmission of characteristics from parents to the offsprings via information encoded biochemically using DNA sequences. It is known as genes. Genetic variations are portions of genetic information at the same genomic site across individuals that can differ between persons with the size of the differing portions ranging from small nucleotide changes to differences extending across the entire chromosomes. The transmission of biological information from parents to the offsprings that encode physiological features has been recognized since early recorded history. For centuries, farmers in various parts of the world employed strategies of selective breeding of crops and animals in order to enhance the desired traits such as crop resistance to an adverse climate, speed in horses, strength in oxen, and large fruits in crops can be accentuated by controlling mating. However, there was no scientific way to predict the outcome of a cross between two particular parents. Dear students, let's now discuss Mendelian inheritance versus non-Mendelian inheritance. It wasn't until 1865 that an Austrian monk named Gregor John Mendel found that individual traits are determined by discrete factors later known as genes, which are inherited from the parents. His rigorous approach transformed agricultural breeding from an art to a science, although Mendel's work was not appreciated immediately. That is why the science of genetics really began with the rediscovery of Mendel's work at the turn of the 20th century by Teschermark, Karl Correns, and Hugo de Vries. And the next 40 years or so saw the elucidation of principles of inheritance and genetic mapping by Morgan. Microbial genetics emerged in the mid-1940s and the role of DNA as the genetic material was firmly established. Mendel's experiments established that genes can exist in alternate forms for each of the 7 traits that he studied, like seed color, seed texture, plant height, flower color, flower position, pod shape, and pod color. Mendel identified 2 alleles, that is, one dominant and other recessive. This discovery suggested a simple functional dichotomy between alleles, as if one allele did nothing and the other everything to determine the phenotype of that organism. However, research early in the 20th century demonstrated this to be an oversimplification. Genes can exist in more than 2 allelic states, and each allele can have a different effect on the phenotype of the organism. So Mendel summarized his findings in 4 postulates, actually in 2 laws, the law of segregation and the law of independent assortment. Let's discuss first the 4 postulates or 2 laws of Mendel. Postulate 1, it was the principle of paired factors. It means that a character is represented in a diploid organism by at least 2 factors. The 2 factors lie on the two homologous chromosomes at the same locus. They may represent the same homologous or alternate expression, that is heterozygous, of the same character. Factors representing the alternate or same form of a character are called alleles or allelomorphs. The second postulate of Mendel was that principle of dominance. When two homozygous individuals with one or more sets of contrasting characters are crossed, the characters which appear in the hybrid of F1 or first filial generation are always the dominant characters, and those do not appear in F1 or first filial generation offsprings are always the recessive characters. Dear students, let us discuss law of segregation. The first law, that is the law of segregation, states that when any individual produces gametes, the copies of a gene separate so that each gamete receives only one copy of that particular gene. A gamete will receive one allele or the other. The direct proof of this was later found when the process of meiosis came to be known. In meiosis, the paternal and maternal chromosomes get separated and the alleles with the same character are segregated into two different gametes. Dear students, let's now discuss the law of independent assortment, which was the fourth postulate, or it was the second law of Mendel. The law of independent assortment, also known as inheritance law, states that alleles of different genes assort independently of one another during gamete formation. But some characters in several organisms do not show Mendelian inheritance, or they show a non-Mendelian inheritance patterns. In such cases, the following characteristic features are observed. One, that there is consistent difference between the results from reciprocal crosses. Generally, only the trait from female parent is transmitted. Second, that in most cases there is no segregation in the second filial generation or F2 generation and subsequent generations. Hence, the non-Mendelian genetics involves the pattern of inheritance that does not follow Mendel's laws. It describes the inheritance of traits linked to single gene on chromosomes. It refers to the inheritance of traits that have a more complex genetic basis than one gene with two alleles and complete dominance. So, non-Mendelian genetics are basically any inheritance patterns that do not follow one or more laws of Mendelian genetics. When scientists began exploring more and more test crosses, they observed that there are several traits that do not match up with Mendel's laws. When scientists began exploring more and more test crosses, they observed that there are several traits that do not match up with Mendel's laws. So, the example of non-Mendelian genetic patterns of inheritance include multiple alleles, incomplete dominance, and codominance. Dear students, let's discuss each one of them separately. Dear students, first the multiple alleles or multiple allelism. The Mendelian concept that genes exist in no more than two allelic states had to be modified when genes with 3 or 4 or more alleles were discovered. When there are more than 2 alleles for a gene in a species, it is called multiple allelism. Dear students, let's first discuss the characteristics of multiple alleles or multiple allelism. Multiple alleles are the alternative forms of the same gene, so they influence the same trait. Multiple alleles of a gene occupy the same locus on the chromosome. Multiple alleles exist at the population level, and an individual level can have only 2 alleles for the given gene. A series of mutant alleles can exist in a population. They are generated by a spontaneous mutation in the wild type and the mutant alleles. Multiple alleles lead to the formation of varied phenotype traits for a character in the population. The wild type allele is mostly dominant over the mutant alleles. The wild type is considered the standard and all other alleles are considered variants. The variant or mutant allele may be dominant, recessive, or has an intermediate phenotypic effect. An individual can have only one type of allele that is homozygous or two different alleles that is heterozygous. Dear students, let us study the ABO blood groups in humans as an example of multiple allelism. A classical example of multiple alleles comes from the study of human blood types. The genes for the ABO blood group are located on chromosome 9 in humans. This blood group was discovered by Carl Landsteiner. The A, B, AB and O blood types are identified by the presence or absence of an antigen A or B on the membrane of RBCs by testing a blood sample with different sera. One serum detects the antigen A and another detects antigen B. When only A antigen is present on the cells, the blood type is A. When only the B antigen is present on the cells, the blood type is B. When both antigens, that is A and B, are present on the cell's membrane, the blood type is AB. And when neither antigen, either A or B, is present on the cell, it is called type O. So blood typing for the A and B antigens is completely independent of blood typing for the M and N antigens. The gene responsible for producing the A and B antigens is denoted by the letter capital I. It has three alleles, IA, IB and small I. The allele IA specifies the production of the antigen A and the allele IB specifies the production of antigen B. However, the allele I does not specify any antigen. Among the 6 possible genotypes, there are 4 distinguishable phenotypes, the A, B, AB and O blood types. In this system, the IA, IB alleles are codominant since each is expressed equally in the IA, IB heterozygotes and the allele I, that is small I, is recessive to both IA and IB alleles. All 3 alleles are found at appreciable frequencies in human populations. Thus, the gene is said to be polymorphic from the Greek words for having many forms. Interpretation of this ABO blood groups in humans is as that there are 6 different genotypes and only 4 phenotypes for blood groups in humans. In AB blood group, that is the universal recipient, the A trait and the B trait appear together in the phenotype. There are 2 genotypes for both A and B blood groups. In either homozygous condition, that is IA or IB or in heterozygous condition, that is IA small I or IB small I with one recessive allele I for O blood groups. So blood group O appears only in homozygous condition, that is small I genotype. Dear students, let us discuss coat color in rabbits is an example of multiple allelism. Another classical example of gene with multiple alleles is the one that controls coat color in rabbits. The color determining gene denoted by the lowercase letter c has 4 alleles, 3 of which are distinguished by a superscript, that is C albino, CH Himalayan, CCH that is chinchilla and C positive that is the wild type actual allele. In homozygous condition, each allele has a characteristic effect on the coat color. Because most rabbits in wild populations are homozygous for wild type C plus allele. The agouti, that is wild type phenotype C plus is expressed as brown fur. The chinchilla phenotype, that is CCH is expressed as black tipped white fur. The Himalayan phenotype CH has black fur on the extremities, that is paws, nose, ears and tails, and white fur elsewhere. Albino or colorless phenotype C genotype is expressed as white fur. So in cases of multiple alleles, dominance hierarchies can exist. In this case, wild type allele, that is C plus is dominant over all other alleles. Chinchilla, that is CCH genotype is incompletely dominant over Himalayan CH genotype. So albino, that is C genotype, and Himalayan, that is CH genotype is dominant over albino C genotype. This hierarchy or allelic series was revealed by observing the phenotypes of each possible heterozygote offspring. This hierarchy or allelic series was revealed by observing the phenotypes of each possible heterozygote offspring. Dear students, let us discuss the eye color in Drosophila as an example of multiple allelism. The eye color in Drosophila, that is the fruit fly or Drosophila melanogaster, is another trait that is determined by multiple alleles. In Drosophila, eye color is controlled by 15 alleles. Wild type or normal allele is W, which produces red eye color, which is dominant over all other alleles. So allele small w codes for white eye color, which is recessive to all other alleles. Alleles of other eye color traits are intermediate between the two, that is dominant red eye color and recessive white eye color. There are a dozen different alleles that show phenotype between the wild type red color, that is W plus, and white color, that is W. For example, coral, blood, eosine, cherry, apricot, etc. Dear students, let's study the other example, that is the duck feather patterns, as an example of multiple allelism. Duck feather patterns, an example of multiple alleles, is at a locus that determines the feather pattern of mallard ducks. One allele, capital M, produces the wild type mallard pattern. A second allele, that is MR capital genotype, produces a different pattern called restricted, and a third allele, small md, produces a pattern termed dusky. In this allelic series, restricted is dominant over mallard and dusky, and mallard is dominant over dusky. In general, the number of genotypes possible will be n bracket start n plus 1 bracket close divided by 2, where n equals the number of different alleles at a locus. Working crosses with multiple alleles is no different from working crosses with two alleles. Mendel's principle of segregation still holds, as shown in the cross between a restricted duck and a mallard duck. Dear students, let's now discuss another non-Mendelian inheritance, that is incomplete dominance. Dominance can be understood in regard to how the phenotype of the heterozygote relates to the phenotypes of the homozygotes. For example, if the flower color potentially ranges from red to white, one homozygous genotype, that is A1 produces red pigment resulting in red flowers. Another genotype, that is A2 produces no pigment resulting in white flower production, where the heterozygote falls in the range of phenotypes determines the type of dominance. If the heterozygote A2 produces the same amount of pigment as the A1 homozygote resulting in red, then the A1 allele displays complete dominance over the A2 allele, that is, red is dominant over white. If, on the other hand, the heterozygote produces no pigment resulting in flowers with the same color as the A2 homozygote white, then the A2 allele is completely dominant and the white is dominant over red. When the heterozygote falls in between the phenotypes of the two homozygotes, dominance is incomplete. With incomplete dominance, the heterozygote need not be exactly intermediate between the two homozygotes. It might be a slightly lighter shade of red or a slightly pink shade of white. As long as the heterozygote's phenotype can be differentiated and falls within the range of the two homozygotes, dominance is incomplete. The formation of a third phenotype, that is specifically with traits that result from the combination of parent alleles, is known as incomplete dominance. Or the incomplete dominance is referred to as intermediate inheritance in terms of trait expression, and none of the alleles from the paired alleles expressed over the other for a specific trait. Dear students, feather color in Andalusian chicken and other example of incomplete dominance. An example of incomplete dominance is feather color in Andalusian chicken. A cross between a homozygous black chicken and a homozygous white chicken produces F1, that is first filial generation of chickens that are gray. If these gray F1 are intercrossed, they produce in F2 generation birds in a ratio of 1, that is black, 2 gray, and 1 white, that is 1:2:1, black versus gray versus white. So the A ratio of 1:2:1, that is the phenotypic ratio, arises in the progeny of a cross between the two parents heterozygous for a character that exhibit incomplete dominance, that is capital A small a is crossed with capital A small a as a heterozygote. The genotypic ratio among these progeny also is 1:2 against a ratio of 3:1 phenotypic typical ratio. When a trait displays incomplete dominance, the genotypic ratios and the phenotypic ratios of the offsprings are the same because each genotype has its own phenotype. The important thing to remember about dominance is that it affects the phenotype that genes produce, but not the way in which genes are inherited from parents to offsprings. The carnation plant, which is an example of incomplete dominance, has true breeding white flowers and true breeding red flowers. A cross between white and red flowering carnation plants may result in offsprings with a phenotype of pink flowers, thereby displaying incomplete dominance. Dear students, 4 o'clock flowering plants are an example of incomplete dominance as well. Red and white flowering plants breed to produce offsprings with pink flowers. Snapdragon also shows incomplete dominance by producing pink colored snapdragon flowers. The cross pollination between red and white snapdragons leads to pink color flowers because none of the alleles white and red is dominant. Dear students, this was all about incomplete dominance. Let's now discuss the phenomena of codominance. Codominance refers to a type of inheritance in which two versions, that is alleles, of the same gene are expressed separately to yield different traits in an individual. That is, instead of one trait being dominant over the other, both traits appear, such as in an animal or plant that has more than one pigment color. Codominance is closely related to incomplete dominance, in which both alleles are simultaneously expressed in the heterozygote. So in codominance, the phenotype of the heterozygote is not intermediate between the phenotypes of the homozygotes. Rather, the heterozygote simultaneously expresses the phenotypes of both homozygotes. We can see as an example of codominance in the Mn blood groups of humans. The Mn locus encodes one of the types of antigens on red blood cells. Unlike antigen foreign to the ABO and Rh blood groups, which also encode red blood cell antigens, foreign Mn antigens do not elicit a strong immunological reaction. Therefore, the Mn blood types are not routinely considered in blood transfusions. At the Mn locus, there are two alleles, the Lm allele which encodes the M antigen and the Ln allele which encodes the N antigen. Homozygotes with genotype LM express the M antigen on their red blood cells and have the M blood type. Heterozygotes, on the other hand, with genotype LN express the N antigen and have the N blood type. So heterozygotes with genotype LN exhibit codominance and express both the M and N antigens. They have blood type MN. Dear students, type AB, blood of the ABO blood type system, is also an example of codominance besides the example of multiple allelism in humans. So in addition to the codominance, human ABO blood groups represent another deviation from Mendelian simplicity, that is multiple allelism, since there are more than two alleles, that is A, B and O, for this particular trait. So ABO blood group is the example of multiple allelism and codominance at population level. The I, A and IB alleles are responsible for the production of A antigen and B antigens respectively. The allele IA for A antigen is codominant with the allele IB of antigen B. Both IA and IB are completely dominant to the allele, that is small I. The hierarchy of dominance relationship is symbolized as IA is equal to IB and is always greater than, that is small I, which is recessive. The red blood cells have antigens on their surface. One of these antigens is the H antigen that is controlled by three alleles, that is IA, IB and small I. Both IA and IB are dominant alleles that when expressed produce enzyme that modify the H antigen. IA particularly adds N-acetyl galactosamine, whereas IB adds galactose to the H antigen. With small genotype alleles, thus lacking the dominant alleles, there would be no such modification on the H antigen on the red blood cells. So a genotype of IA or IA small I would result in H antigen with N-acetyl galactosamine on the red blood cells, a trait indicating type A blood. A genotype of IB or IB small I would result in H antigen with galactose, a trait indicating type B blood. A genotype of IA IB would result in H antigen with both N-acetyl galactosamine and galactose. This indicates type AB blood. Conversely, a genotype of small I which is recessive means lacking such modification on the H antigen and an indication of a type O blood, which is devoid of any antigen on the RBC surface. In codominance, blood type AB implies that both dominant alleles are present and expressed together. Other examples of codominance include that speckled chicken has alleles for both black and white feathers, and roan cattle which express alleles for both red hair and white hair are examples of codominance. So codominance is exemplified by a plant rhododendrons that bears flowers with two distinct color phenotypes. For instance, a white spotted red flower could be caused by a cross between a red flower and a white flower. So the alleles for the red color and white color phenotypes mean that they are codominant with each other. Dear students, it was all about today's lecture regarding non-Mendelian inheritance, multiple allelism, incomplete dominance, and codominance. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.